So, like we just said, heterozygotes are not true breeding. So see if you can figure that one out. Think about it. I'm going to ask you when we go back to class. So, because sometimes dominant traits cover recessive traits, we can't always tell an organism's genes just by looking at it. So we have two different ways to distinguish an organism. We have phenotype and genotype. Phenotype is the physical characteristic that we're looking at. So if we're looking at flowers, there's two phenotypes, purple and white. This is different than genotypes, which is the actual genetic makeup. So it's pretty easy to remember. Phenotypes is physical, genotypes is genes. So if you have purple flowers, that's your phenotype. You are purple, but you could have two possible genotypes, big P, or big P, little p. So that's why it's important to distinguish between the two. So this guy right here, his genotype is big P. He is homozygous. He has two copies of the same dominant allele, so we call him homozygous dominant. His genotype is big P. His phenotype is purple. These two guys, their phenotype is also purple, but their genotype is big P, little p, so they're both heterozygous. This white guy down here, his phenotype is white, but his genotype is little p, so he is homozygous recessive. And you can see that the ratios are important also. So remember that Mendel saw ratios of 3:1 for dominant to recessive phenotype. And that's what we're usually going to see. But that's different than the genotypic ratio, which here is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. So the genotypic and the phenotypic ratios are not always going to be the same. So it's really important to know, how can we determine what a dominant phenotype's genotype is? Right? If you have a purple flower, are you ever going to know if it's purple or if it's big P, or little p? How could you know? Right? So the best way to do this is to perform a test cross. And what this means is taking your unknown, your purple flower that you don't know what it is, and crossing it with a true breeding homozygous recessive individual. So you would take your purple flower that you don't know if it's homozygous dominant or heterozygous, and cross it with a white flower. Because you know that person's genotype, that flower's genotype, it's going to be little p. And so you can tell by the offspring what the genotype of the parent is. So here's our unknown guy, our unknown purple. Is it big P or big P, little p? We don't know. We're crossing it with a white, and we know this is little p. So when you cross them, we'll get two possible outcomes. Either all of your offspring will be purple, or half of them will be purple and half of them will be white. If they're all purple, you know that you had a big P parent, or it's likely that you had a big P parent. If you have if you get some white offspring and some purple offspring, then you had a big P, little p parent, because there's no way you could get white offspring unless there was unless you get a recessive trait from both parents. So there must have been a recessive trait in this parent. So that's really important. Now I told you Mendel had two laws. The first one was the law of segregation, which is when the alleles separate during meiosis. So the two alleles that your parents have are not going to be necessarily what you got. And the second one is independent assortment. We talked about this in meiosis, so you should be a little bit familiar with this. Okay, what this is talking about is when we look at one trait like we did right here, this is called a monohybrid cross. Remember, mono means one, and we're looking at crossing two different kinds of things, purple and white, so it's a hybrid. So this is a monohybrid cross. We can do dihybrid crosses when we look at two traits together. So for example, right here, we're looking at two different traits, not just one. This seed is yellow and this seed is green, but this seed is also round and the seed is wrinkled. So we're looking at two different traits instead of just one. This is a dihybrid cross. Okay? So read in your textbook about how to complete that. So looking at a dihybrid cross, Mendel was able to determine the law of independent assortment. This is really important. The law of independent assortment says that the alleles segregate independently of each other. So just because you are yellow and round does not mean that all of your gametes are going to be yellow and round, okay? Your yellowness and your roundness segregate independently of each other. So this is where we get genetic variation from, like we talked about with meiosis. Now, this only applies to genes that are on different chromosomes. So Mendel was really, really, really lucky that all of his 7 traits were on different chromosomes, so it was never affected. Because think of it this way. You know that the genes are on the chromosomes, so if we're separating the chromosomes, if you're yellow on the top of this chromosome and round on the top of this chromosome, then every single gamete is going to be round and yellow. If if they're on the same chromosome. But if they're not on the same chromosome, then they're always going to assort independently. And that is it for 14.1.